Right, we're now getting to the nitty-gritty when the careful use and practice of micropipetting is going to pay dividends. You'll see that in our very homemade version of a test tube rack, using a little piece of sponge to hold these tiny little capsules, that's the amount of liquid that we're dealing with. And this is our stock sample. So we're going to subsample 7.5 microliters from here into a different tube to go further into our DNA extraction process and amplification later. So Mary, if you'd be kind enough to lead us through the practicalities of how we do that, that will be very helpful.
Okay. Now, these little tubes are called, they've got various names, Eppendorf's, microcentrifuge tubes, etc. Most important thing to remember is that they are supplied as far as possible free of DNA, and they must be kept free of DNA, of any foreign DNA, in use. So we never have the caps open until we actually are ready to transfer.
We certainly don't want our fingertips all over the edges, do we, in any way?
No, and certainly don't want or hairs or anything going in. It's also important to keep the tips as free as possible of any extraneous DNA. It might be sensible to put them in a box like this, but you can just have them in a clean plastic cup and just make sure that you take them out very carefully. And it may be sensible to wear gloves, just simple, ordinary plastic gloves. It's the DNA that matters, not, microbes and things like that. Now, to transfer 7.5 microliters, you twiddle the knob at the end of the micropipette until it reads exactly 7.5. You can see on this micropipette there is a white decimal point.
And we're now seeing the benefit of having a pipette that you can adjust the volumes to.
Absolutely.
Yeah, for different recipes, for want of a better phrase.
That's right, for different activities.
Yeah.
What you now do is you put a tip on without touching the tip. And you take up the sample that you want to transfer. So you flip the cap off, push the air out.
So that's the soft stage you mentioned earlier.
Absolutely, the soft start. And then the tip goes just underneath the surface of the liquid, and then you release from the soft start.
Okay, now we can see our sample that we're after right at the tip of the pipette.
Absolutely. The material, the liquid, is going into this empty Eppendorf, and I'm going to go right down to the bottom, and I'm going to touch the surface of the plastic, and I now go all the way down to the hard stop. Now, holding the plunger on the hard stop, I now gently and slowly remove the tip.
Okay, and we'll see then. Tip is free of any liquid, but no doubt around the surface there might be some microscopic amounts, and hence why we can't use that tip again.
That's right.
Yeah, okay.
So that tip is now contaminated with that DNA, so discard it.
Okay, and I think it would be worth some wet runs with students, just practicing that with any coloured dye, so they can see what's happening really before they go on to
anything at all.
And
you may want your students to actually take 7.5 microliters of that drop and put it into another tube.
Yeah, okay.
You may also choose perhaps to use coloured tubes with your students. It may make the activities easier.
Because in terms of contrast, is to see the colours?
It's so that they don't mix up the samples.
Oh, sorry, okay.
Because the samples may all look the same.
Right, okay.
And it would help, when you're presenting the class, presenting the activities to students who are not experienced, to say, transfer
use
from
the green one.
tube
Yes,
to the
I can
tube.
see that would help. Certainly help me even. Transfer from green to blue or vice versa. Yes.
Yeah.
Okay. Right. Thank you. I think we have that section covered.